All right, welcome to our continuing macromolecules series, part 4 of the end with nucleic acids. So, nucleic acids are our final category of macromolecules, and the monomer of a nucleic acid is a nucleotide. The polymer, amazingly enough, a nucleic acid. The bond that holds monomers together is a phosphodiester bond. Nucleotides are built out of 3 basic ingredients, right? There's a carbohydrate, a phosphate functional group, and a nitrogenous base. The carbohydrate is based on ribose, so it's a, to be most specific, 5-carbon sugar. And it comes in 2 flavors: ribose or deoxyribose. If you look at ribose as chemical, we got our 1, 2, 3, 4, 5 carbons. And then we've got a bunch of OHs and Hs on it. And there we go. So this is ribose. It's not too terribly complicated. And if we want to know how ribose differs from deoxyribose, it's actually very simple, because deoxyribose is ribose missing something. Deoxygenated. And if you want to know how to turn ribose into deoxyribose, all you got to do is deoxygenate 1 oxygen off of there. And now you have deoxyribose. So, carbon numbering is actually rather important when looking at the 5-carbon sugar. So we're going to talk carbon numbering in these sugars. It is given a little apostrophe next to the number. That's prime. So 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. All right, so that's our sugar. Next up is the phosphate group. That's pretty easy. Phosphate as a functional group is OPO2, 2 minus. So it will be the functional group that is the site of the phosphodiester bonds between nucleotides. Lastly, we have nitrogenous bases, and there are 4 flavors of nitrogenous base in DNA, 4 flavors of nitrogenous base in RNA, but conveniently enough, DNA and RNA share 3 identical nitrogenous bases. So you're not memorizing 8 nitrogenous bases, you're memorizing 4, and then the unique one in RNA. So speaking of DNA, RNA, right? The naming scheme is pretty easy. NA is for nucleic acid, and then the D or the R tells you which 5-carbon sugar it has. DNA is deoxyribonucleic acid. RNA is ribonucleic acid. not too terrible. So again, just looking at how we have a loss of an oxygen on that 2' carbon. You don't have to know that it's the 2' carbon, really just that there's one less oxygen on deoxyribose. The phosphate group. It is charged, and with all those oxygens on there delivering their electronegativity, it has a partial negative charge. So, phosphate groups have a negative charge. And this means when you link up a bunch of nucleotides together into that polymer, you're going to have a negatively charged structure. Okay, so the sugar phosphate backbone is what forms when you have the bonds that hold nucleotides together. Why is it called the sugar phosphate backbone? Simply because it's literally phosphates holding together sugars. So if we look here, we'll have our ribose. We'll throw a nitrogenous base here. We'll just make it a circle. And then there's our 5 carbons. Now, we'll remember our numbering. 1', 2', 3', 4', 5'. And then we'll put in a phosphate group. So, one of the universal rules about binding to hold these nucleotides together is that a phosphate always connects the 3' carbon of a carbohydrate to the 5' carbon of the next one down. So it's always a bond between a 3' and a 5'. So that is actually rather important because, interestingly enough, when you have DNA, it is a double-stranded chemical, which means for every nucleotide on one side, there's going to be a nucleotide paired up with it. So there's some nitrogenous bases. So DNA is double-stranded. And essentially, it's held together by hydrogen bonds between the base pairs. Not too terrible. What does that look like? Well, boop, 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 boop. Let's make this a circle. And then this one will be a square. And then the triangle can hydrogen bond to, I don't know, man, let's just make it like a hexagon. There we go. Okay, so we have hydrogen bonding between base pairs to hold the two strands together. Here's one nitrogenous base, here's the other, so we have hydrogen bonding between base pairs. Now, the other interesting thing about this is that these are anti-parallel. Parallel simply meaning that, I'm pretty sure I spelled parallel wrong. Let's try it with 2 L's and 1 L. It doesn't look right. Anyway, parallel because they go alongside each other. Anti-parallel because they actually have opposite directions, right? So if you look at the DNA strand that I have done so far, there's a 5' carbon at one end, and then at the very end here, there's a 3' carbon. So there's direction here. You could say that this strand is in the 5' to 3' direction. Whereas the anti-parallel strand is going to be going in the opposite direction. Let's get these on there. And one more. All right, and now let's go ahead and put some phosphate bonds to connect these guys. Well, phosphodiester bonds. I said phosphate bonds, but phosphodiester bonds. All right, so there we go. This is anti-parallel. It's running in the opposite direction. So if you look at one end, we see this terminal end here with the 3' and this terminal end down here with the 5'. And so this strand is running in the 3' to 5' direction. So that's how these work. We have hydrogen bonds between nitrogenous bases holding together our DNA strands. And the strong covalent bonding between nucleotides is just between sugar, phosphate, sugar, phosphate, sugar, and phosphate. So this is the sugar, the sugar, oh my goodness, stop using a search thing. Oh my gosh. Apologies. Sugar, phosphate, backbone. So, sugar phosphate backbone simply because phosphodiester bonds bind two 5-carbon sugars together. So, sugar, phosphate, sugar, phosphate, sugar, phosphate, and so on. So, sugar phosphate backbone is the strong covalent bonds holding together the strand of DNA. Whereas when you have the double-stranded structure, hydrogen bonds between the base pairs are what keep the double strands connected together. So, that's pretty cool. Okay. Okay, so that's the sugar phosphate backbone with the phosphodiester bonds. And now let's talk nitrogenous bases. Nitrogenous bases can be separated out into purines and pyrimidines. Purines are double ring nitrogenous bases. These are adenine and guanine. Pyrimidine nitrogenous bases are single rings, and these are cytosine and thymine. Now this is my DNA list. Oof. I don't know if the recording is picking it up, but apparently there's like a little search box in the Microsoft Office or Microsoft PowerPoint thing, and even though I'm in a presentation, apparently if I click in this general direction, it'll go to the search box. Okay, so DNA's nitrogenous bases are adenine, guanine, cytosine, thymine. With cytosine and thymine being the pyrimidines, and adenine and guanine being the purines. And the purines being a double ring. Whereas the pyrimidines are a single ring. Okay. I did mention that RNA has an odd one out. So, for RNA, adenine is the same, guanine is the same, cytosine is the same. The only difference in nitrogenous bases comes with the pyrimidine uracil. So, both DNA and RNA have adenine, guanine, and cytosine. DNA has thymine, RNA has uracil. Ta-da! Not too terrible. Now, the nitrogenous bases hydrogen bond together in a very specific pattern. So to get those hydrogen bonds between nitrogenous bases, in order to have those double strands held together, you have hydrogen bonding. And there's a set of basically this always hydrogen bonds to this called the base pair rules. So to talk about the base pair rules, first, you see I put AGC over here on there. We're going to run with these as our abbreviations for our nitrogenous bases. AGCT and AGCU. If you see U or uracil, you know you're talking about RNA. Okay. So, hydrogen bonding is pretty simple. We call it the base pair rules. And it's surprisingly easy, right? A always hydrogen bonds to T. G always hydrogen bonds to C. And where A would hydrogen bond to T in RNA, it's A to U. All right, and vice versa. So if I have a strand over here, and it's got A, T, T, C, G, T. There we go. I can automatically know what nucleotides are in the complementary strand, the opposite strand, and that's going to just be their base pair. A to T, T to A, T to A, C to G, G to C, T to A. And there we go. I know easily that system. So it's pretty simple. We have our double ring, single ring, purine, pyrimidine. We have our adenine, guanine, cytosine, which is universal between DNA and RNA. And then we have thymine and DNA, uracil and RNA. And then the base pair rules are A to T, G to C, and A to U. Not bad. So, this was a terrible joke. All right. This is just looking at each of them. So, here's uracil right here. And we can see the OH right there, which means that's ribose. Which means this is RNA, so you can believe my bullet that already told you it's RNA. Boop, boop. Alright, the base pair rules. We call the base pairs complementary to each other because A always to T, G always to C, which means we also have complementary bases and complementary strands. In other words, those anti-parallel DNA strands are complementary. Whoa, hey, no. A to T or U, there we go. Deoxyribonucleic acid, blah, 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 blah. Double-stranded, anti-parallel, right? Complementary strands that are held together by hydrogen bonds between base pairs. They run in the 3 to 5 and 5 to 3 direction. And lastly, they have a helical shape. What does that look like? You've probably seen it before. Draw a ladder and then twist your ladder. It's a twisted ladder. So it's always drawn like this, where the little bars or rungs in the ladder represent the hydrogen bonds between base pairs. And there we go. We have our double helix. We have our hydrogen bonds between base pairs. Right? We have our complementary anti-parallel strands. And anti-parallel again because one's going 3 to 5 and the other's going 5 to 3. Not too bad. There's a picture that kind of puts things together. Obviously not as great as the art that I've been using. But you also notice with those base pairing rules, A to T, G to C, it's also purine to pyrimidine. So the purine adenine will hydrogen bond to the pyrimidine thymine. So also kind of interesting. So you can see we have our 5. Whoa, stupid search box. We have our 5' and 3' end. And phosphate is always bonded to the 5' carbon, and then connects a 5' carbon to a 3' carbon. All right, so here's a 3' carbon. And there's a 5' carbon. Not too terrible. So 5 to 3 because the 5' end is the terminal end there. And the 3' end is the opposite terminal end because these are linear strands. So they have a beginning and an end. 3 to 5 on this one. And there you go. Okay. RNA is ribonucleic acid. Big thing about ribonucleic acid is that it is single-stranded. So it is not naturally forming a double helix because it is single-stranded, right? So no double helixes. It's a single-stranded structure. Again, AGCU, A to U, G to C. And it has a number of functions. So DNA's main function is genes. So as far as functional differences, DNA is your genes or genetic storage. RNA is actually pretty diverse as far as function in a cell. There's a type of RNA called messenger RNA. Messenger RNA basically takes instructions for protein from the nucleus to the cellular machinery where protein gets made. So mRNA, right, copies a gene into mRNA and then sends it to get the protein made, right? So it goes nucleus to ribosome. That's going to be coming up in the next lecture, so might as well give it a mention here. Ribosomes are the site of protein synthesis. So mRNA copies a gene from DNA into RNA, specifically mRNA, and then it travels from the nucleus to the ribosome, and then it's our protein blueprint, because all genes code for a protein. So, not too bad. Ribosomes are the site of protein synthesis. So, amazingly enough, ribosomes involved in protein synthesis. Again, so ribosomal RNA or rRNA. So, not too bad. Again, ribosomal RNA is the main component of a ribosome. Major ribosome component. And that's, again, ribosomes are the site of protein synthesis. So, not too terrible there. And then, lastly, proteins, as we learned in the protein section, are built out of amino acids. Amino acids are not necessarily synthesized right next to a ribosome, which means if you have your little ribosome here, right, and then you have your mRNA, which carries the ingredients or the instructions on how to make the protein, you also need to bring amino acids in order to actually make the protein. So tRNA is transfer RNA, amazingly enough. Who'd have thunk it? And transfer RNA transfers amino acids to the ribosome. So once more, we're all about protein synthesis. Transfer RNA kind of got blocked up there, but it's abbreviated as tRNA. So now you have mRNA, rRNA, tRNA. And we can see just how intimately RNA is associated with protein synthesis. It's the blueprint for making a protein. It's also making up significant portions of the actual site where protein is made, and a type of RNA is gathering amino acids and bringing it to the chain so that you can actually grow a protein, a polypeptide, into a protein. This is just a picture of RNA showing the C to G and A to U. It does not normally loop on itself. This is for demonstration purposes. All right. Special mention to ATP. ATP is not technically a nucleotide, a nucleic acid. And that's simply because there's no phosphodiester bonds, right? We don't have a sugar phosphate backbone. ATP is a pretty simple chemical. Adenosine triphosphate. Now, with adenine on there, you might be able to guess which nitrogenous base adenosine triphosphate carries. And if you said adenine, congratulations, you're correct. So, we'll just put an adenine on here. All right. Again, we have our 1, 2, 3, 4, 5 carbon sugar, and it is ribose. And connected to that ribose are 3 phosphate groups. So it has similar ingredients, right? A nitrogenous base, a 5 carbon sugar, and phosphate. But rather than having like a phosphate backbone, a sugar phosphate backbone or anything like that, what we have is 3 phosphate molecules with their negative charge chained together. Like charges repel, so these are trying to push each other apart. In addition, the bonds between these are called phosphate bonds, and they are extremely weak. Weaker than hydrogen bonds. Hydrogen bonds are a non-covalent attraction between hydrogen and something electronegative. And this is even weaker than just a non-covalent attraction. So this is kind of a big deal here. They are trying to yank each other apart, and they are held together by a very weak bond, which means this whole phosphate section here is extremely unstable. But because it's unstable, that means there's a bunch of energy just waiting to burst out. And so you can knock a phosphate off, and when you do that, the phosphate will get broken loose, and you'll also get a release of energy. ATP on its own is just an energy sink. It's something nice and unstable, so you can pack a lot of energy into it. You don't get a lot of lost energy to making random other chemicals to store energy worse than ATP. So, the cell builds a bunch of unstable ATP so that the ATP can be shuffled around to places where it's needed, and then boom, pop off a phosphate, and you've got the energy that was stored in that bond. Ta-da!